This is a video to explain the second lab that we're completing from our Carolina biological kit. It's the isolation streak plate method. So you're going to be taking a mixture of bacteria and streaking them onto an agar plate with the hopes of getting isolated colonies. Before you start, it's important to make sure you have everything you need. To complete this, you're going to need two bacteria that you have reactivated. So Serratia, we reactivated for the first lab, so you should still have that. I instruct you to save that culture. And so you're also going to need to activate the Micrococcus culture. So that's going to need some time to grow until you see turbidity. So if you haven't activated the Micrococcus, you need to do so. And then once that has been activated and grown, you'll need the rest of the equipment that's listed here, including a sterile plastic culture tube, transfer pipettes, and inoculating loops. So now that you are sure that you have everything you need, make sure that you do these things before you start each activity. Make a bleach solution that's used to disinfect surfaces. Again, this is done before and after you perform an activity. And get your isopropanol container ready for disposal of tools. And of course, always wear the appropriate PPE. So the first thing that we're going to do is make sure we have our activated cultures. So I have Serratia and also Micrococcus. These are the two bacteria that you need. And this is an empty test tube culture tube that we're going to use. So because we're going to be opening glass tubes, we're going to get our flame ready to flame tubes when we open them as well as when we close them. I'm opening a transfer pipette that I'm going to transfer the first bacteria. This is Micrococcus. So before you use it, it's helped to do a brief mix. And so when you open the tube, you'll pass the mouth through the flame, and then you're going to draw up some of this liquid culture. You're going to add 10 drops to the sterile culture tube. So flame the tube before you close it, and then to the sterile tube, don't flame this since it's plastic, add 10 drops. It doesn't have to be precise if you add too many, if you add 11, but make sure you get at least 10 drops. And then you'll cap that. I'm going to put the rest of this liquid back in here, so I'll flame the tube and add the liquid back in there. And of course, flame the tube before closing. And because I'm done with this transfer pipette, it goes in the isopropyl alcohol disposal. And so then I'm going to use a fresh transfer pipette and get some of the other bacterial culture. And so this is the Serratia culture. This is just going to, you just need a little bit of this. And so I'm just trying to suck up a small amount into this pipette. That's too much, so I'm going to add some back. And that's still not perfect. So you just need a little bit. You're going to add 1 to 2 drops of this into that same plastic culture tube. So just a couple drops, 1 or 2 is fine. And then you'll discard that, that transfer pipette as well. We're done with the glass tube, so we can extinguish our candle. So I'm going to get a sterile loop out, an inoculating loop. So these are the plastic inoculating loops that we've used before. And the sample that we're going to use is the mixture that we just made. So that's, again, a mixture of Micrococcus and Serratia. And so I'm going to get a loop full of that. And on my plate, I'm going to streak just one area with this bacteria. So I'm going to flip the plate over and then just streak one area, moving in a back and forth zigzag manner. And then I'm going to discard that loop. Now I'm going to get a fresh loop, and I'm going to overlap a little bit and pull some of the bacteria that I just placed into the next zigzag zone. So that's the second zone. Discard that loop. I'm going to get a third loop and repeat this, pulling the bacteria from the second zone into a clear area on the plate. So you kind of have to know where you've been previously. So that's the third area. And then I'll repeat this one more time with the fourth and final loop. So again, I'm just going to come in contact with bacteria from the third zone and pull it out into a free zone, the fourth zone that doesn't have any bacteria on it. So this is known as a streak plate for isolation, and the hope is to take that mixture of bacteria that we had in the plastic tube and spread it over the surface of the plate to get isolated colonies. So the next steps include making sure that you disinfect the tools you used in the alcohol for at least 30 minutes. The one culture that you want to save is the reactivated micrococcus broth, because we'll need that in the next lab activity, so make sure you hold on to that. Just leave that out. You can leave the culture tube with the lid slightly loosened, and it'll be good for another 10 days to 2 weeks. All the Serratia cultures you no longer need, so those can be properly disinfected and disposed. Remember, those need to soak in a 10% bleach solution for 24 hours prior to discarding them. And then the plate that you just set up, this streak plate for isolation, incubate that upside down, and you're going to take photos after 48, 72, and 96 hours. So when taking those photos, remember to include a post-it note with your name and the date, and try to take photos in such a way that I can see the growth well on the actual Petri dish. So I just wanted to quickly show you a streak plate for isolation that I performed in this experiment. Most importantly is that you can see two discrete, different, isolated colonies in that final zone, so kind of the top part of this picture. There are the Serratia colonies that have their reddish pigment to them, and then the Micrococcus is going to be yellowish in color. And so there are two different types of colonies that can be visualized here. And that's the goal of this. Remember, a streak plate for isolation is a technique that allows you to go from a mixture of different bacteria. And so we mix together the Serratia along with the Micrococcus in a single tube. And so they were mixed together. And then by doing this technique called the streak plate for isolation, by the final zone, you have individual colonies forming. And so that allows us to have isolated colonies that we can then grow in pure culture if we wanted.